We also support electric propulsion research. We are also building instruments for space and some other ISRO related activities our lab is supporting. So I'll quickly go through the other activities first for 2-3 slides. So this is the kind of support we provide for the ISRO's electric propulsion, that is plasma propulsion project, where our main responsibility is to provide the diagnostics. So we build the diagnostic tools, we test them, calibrate them in our lab, and then we supply it to the ISRO facility where the testing is done day to day. Our students get an opportunity to, of course, spend time with these testing facilities. Since we were building instruments, we started building some instruments for space also. So we have been sending probes to study the ionosphere of Earth. 2019 was our first mission, 2023 was our next mission, and in coming years we are going now next to IDM mission, which is this year sometime, and then RPAV, that is a Venus mission. We will be part of the Venus mission also. So basically we build instruments, we do mass spectroscopy, and we do some science. So I will be now coming to the science. So I don't think this community needs to be told. So this is just one slide about the various type of systems where we do see a lot of organic matter in our own solar system. Comets, dwarf planets, asteroids, planets, and moons as well. What is shown this side is some of the examples. This is the sample collection from Bennu, which is again rich with carbonaceous substances. This is Titan. This is the actual image taken by the Huygens lander, and all this yellow and brown material what you see is basically organic matter. This is Pluto, which is practically fully covered with either nitrogen ice or organic matter. So it is something which is very commonly present in our solar system, something which was not expected say 30-35 years back. So as a molecular physics person, we divide it into few categories, and this is purely from a molecular physics perspective. One is interstellar medium, where the densities are really very low and the temperatures are also very low. So when it comes to studying the molecular dynamics, we have to keep that in mind. When it comes to protoplanetary disk or molecular cloud, the densities are higher, not very high, but say a few orders of magnitude higher than what I would have in ISM. It can be cold, it can be hot, because there is a star in the center which is giving out energy. It produces asteroids and comets which in the later stages of the solar system evolution remain as the fingerprints of what happened in the beginning, and it is also mainly driven by the stellar radiation and cosmic radiation. Then the third category is the planetary upper atmosphere. So planetary upper atmosphere is largely driven by solar radiation directly. Then there are lots of secondary reactions which are possible. Excuse me, I think. How to go back in this?
Yeah, the other. Yeah.
Okay. Yeah, sorry. So, and they are of course affected by local factors like gravity, magnetic field of the system which is holding. Of course, the question would be, why not atmosphere at large? Again, coming to molecular physics perspective, the moment it comes to dense atmosphere, millibars or bars of atmosphere, simply a local chemistry takes over compared to what high energy radiation would do to the molecules, and that is the main interest we have. So we focus mainly on the upper atmosphere, ionosphere, that part. This is a view not from a molecular physics perspective, but more like a technician's perspective, like what techniques I need to worry about and how much money I have to pay for it. So one is, again, sorry, this I'm still not used to this very advanced equipment. So this in situ measurement is something which we all like, but the problem is something has to go to some planet or moon or somewhere, land, and do the measurements and send the data back. Very challenging. It has usually very long lead time, and of course the scope is very limited. You can't just go there and say, oh, my mass range is up to 1000, but it would be nice if it can be made 2000. You can't do that because the instrument is already sent there. The other is sample return. That is fantastic because then it is in your lab, you can do whatever you want, you discover something new, you do another investigation. But then the problem is a sample return mission is an order of magnitude more complexity than simply doing an in situ measurement. But it has been done. Like this is one picture. This is December 2020. Hayabusa 2 sent back a small capsule, just this much size, which got only 5.5 grams of an asteroid piece, Ryugu, and it actually changed a lot of our perception about how the solar system evolved. Just that 5.5 gram sample, some picture, representative picture I have given here. Observational, fantastic. You can keep building facilities, keep building telescopes, keep doing observations. Problem, you can only see what you can see. For example, those elements or those molecules which can emit or fluoresce, you can see. Those don't, you can't. And that sometimes becomes a problem for molecular physics. It is nice to sort of mimic what would be happening in that particular environment, but again, it's a lot of guesswork, a lot of modeling work. It's not something very easy. You can see everything in the lab, but well, it may not be there. We don't know. So combine the molecular physics perspective and the perspective of a machinery person who would like to, put money, build instruments. This is big conflict. Okay, so then we come to, then why should we do laboratory astrophysics or astrochemistry, if you want to call it that way. So these are few philosophical reasons why we should do it. One thing is, it is very clear there is much more organic matter outside our planet than on our planet. So that's very clear. We are not interested in what happens in the atmosphere at very high densities where the reactions can happen, like I am breathing air, a lot of chemical reactions is happening. Solar radiation has very little to do with it. So from a molecular physics point of view, I learn very little. So doing it in the lab in vacuum is very nice for us. There are too many commonalities. For some very strange reasons, certain molecules which are biologically important seem to be present in an environment which was existing before the planets were even formed. That's what was proven by Ryugu. For example, that some of the amino acids existed before the first planet was formed in our solar system. So why it is like that? Another very interesting problem is the handedness. Our life is left-handed. Why it is left-handed? And somehow the molecules in space also seem to have certain tendency. We want to understand that. So it is important to do that in the lab. Observations show you the status of what exists. What they don't tell you is how the situation reached to that status. And that you can only understand when you combine what you observe with what you do in the lab. So that is another reason why one should do laboratory astrophysics. This is more practical approach why one should do. Observations are limited in many ways. In situ sample return opportunities or in situ or sample return opportunities, as I said, are very limited. There may be lessons to learn. These problems in astrophysics and astrochemistry can become very important problems in fundamental molecular physics, and I will show you a couple of examples of that. And then fantastic ground for training new generation in various techniques, whether it is science or technology. It has a lot of spin-offs. Then it helps us a business case for future expeditions. So this is also another very important reason why one should do laboratory astrophysics. And again, what we can do, this is a list of things one can do. You can do spectral characterization, you can develop a mechanistic understanding of how different molecular processes are happening, etc. I'm just speeding up now. And then at the end, the spin-offs. It has a lot of interesting spin-offs. I could discuss with you how we are now actually, what we developed for astrochemistry is now how it is going to industry and so on. I can discuss that also with you. So what do? We basically study energetic radiation, how it interacts with organic molecules, especially aromatic molecules in space. So we try to mimic that, we try to understand that. Among the various types of processes you would like to study, we focus mainly on the statistical processes, and we also try to do some ionospheric simulation like Titan ionospheric simulation. How do we do it? We use mainly mass spectroscopy and coincidence spectroscopy for these experiments. We use VUV radiation, UV radiation, charged particle radiation. We look at fundamental processes like plasma on resonance, isomerization and dissociation, And very recently we have discovered something really nice, intracluster ion molecule reaction, which is very important for the evolution of organics in protoplanetary disk. And the main foundation of our approach in all these experiments is why, the last line, we are trying to propagate or sort of find support for a top-down approach. The by default approach of evolution of larger molecules is you start with smaller molecules. We are trying to project something which is upside down, not something which is unheard of in astronomy. For PAHs, we understand that pH evolution now is going top down, but even organics in general we are proposing top down. Now why we are proposing top down approach, I'll just give you three examples why we believe, but there are I'm sure many of you know already a lot of these examples. What I have on the left hand side is the ratio of hydrogen isocyanide to hydrogen cyanide as a function of heliocentric distance. And as you can see that these are for comets, a large number of comets, and you can see that it is actually just going down by a very specific way. We can again get into the details, but I am just skipping all the discussion and saying this is essentially due to photodissociation of large organic nitrogenated aromatics. Why, how, we can discuss. You can, sorry for the size of the fonts, the reference is given to you. Another example what you have is a schematic here for how the ionosphere of Titan behaves. Titan is the moon of Saturn, and it has a lot of organic matter. It has atmosphere, dense atmosphere, and there again there are signatures that the astrochemistry or photochemistry there is driven by a top-down approach and not bottom-up approach. The last is a protoplanetary disk. Again, the question is, should it be top-down or bottom-up? Bottom-up sounds very easy because you have a cloud of gases roaming around a very energetic star. Excuse me, something is happening, okay. We sit at 1 astronomical unit, and by the evolution of protoplanetary disk, actually the organic matter should have condensed beyond 10 astronomical units roughly. But we have it here, so that means there are mechanisms which are bringing that material down here, and if you go into the details of that, we start discovering the mechanism should be more from top to bottom. Again, I am saving time and moving ahead. We can discuss why and how. So what do? We take protons ranging from 10 to 150 keV from an ECR ion source. We take photons. At IIST we use a multiphoton, we use a tunable OPO to get nanosecond pulses of laser. We also go to synchrotron radiation sources where we can get extreme UV radiation ranging from 80 eV to hundreds of eV in principle. We also use electron beam for our experiments. We mainly look at statistical processes which are dominant in PAHs and nitrogenated PAHs, and we try to recreate the situation in the lab. The main problem is there are literally thousands and thousands of these molecules. You can't simply keep taking one molecule after the other and doing this. So we are trying to look for a universality in this process, okay, and this is the work which we have been doing. And what is this universality? The universality is in basically every time you excite this PAH or PANH, it undergoes some structural changes, isomerization and then dissociation, and this pattern actually is a complex mix of isomerization and dissociation and then reaction, which actually leads to the evolution of the material there. So we try to understand if there are universalities in this. You can find the summary of what all universalities so far we have found, and now recently we found something more. This is nicely summarized in this AstroPH volume. We use mainly gas phase beamline in ELATRA, Trieste. We also use LEIBF at IUAC New Delhi, low energy ion beam facility. We have a very interesting ion neutral coincidence energy correlated time of flight mass spectrometer which is built at IIST at our lab. And we also have a reflectron time of flight with electron impact. So this is how the system today looks. We have a source of ions, electrospray ion source and ion trap, And then we have a parallel plate analyzer with a energy correlated time of flight. I will show you some of this data. I am just not going into the instrument that much. Just to highlight, these instruments are all built at IIST by our PhD students. So this is an ion trap which was built by my latest PhD student, and she did all the electronics, and she built the electronics by hand and made the trap work. This is another master student. She did this job. She built the Titan ionospheric reactor at IIST. So this is our Titan reactor setup. I'll skip that part. So let us come back to the physics, because instead of talking more about instrument, I would like to now go into details of the physics for next 10 minutes or so. So we are talking about proton, photons, and XUV or EUV photons, and we are talking about plasmon excitation. So this is the only slide where I am going to talk about plasmon excitation and move forward to the dissociative processes. So why plasmon? Because look at the cross section. So what is plasmon? Is basically once you have a delocalized electron cloud on an organic molecule like say benzene for example, and you excite it, there are certain modes where the entire electronic cloud, delocalized electronic cloud, oscillates against the molecular cage. And this kind of oscillation is universal. It's not like benzene has one, naphthalene has another. It's actually for all PAHs it is 17 electron volts. All PANH also it is 17 electron volts, and you can see this is the photon energy of excitation, and the cross section, you can see the cross sections go in hundreds of megabarns, and this is only photo excitation. Usually charged particle cross sections are this high, but this is photon induced. Charged particles are even higher cross section of excitation. For some reason I don't find this particular phenomena included in astronomical models, but my knowledge is very limited about it. If somebody knows better, please give me some advice. But definitely this is something which exceeds all other standard photoionization processes by orders of magnitude, and this we are not including in many of the models, so of course there is something not complete. Our group is at present one of the only two groups in the world where we study explicitly plasmon excitations. The other group is a TIFR group where I come from, where I was trained, where plasmon excitation in PAHs and PANHs we study. Excuse me. Yeah, so what are the molecules we have been studying? These are just the example molecules. As I said, there are really thousands of molecules. So we study naphthalene, azulene, anthracene, pyrene, fluorine, quinoline, isoquinoline. The first line is basically PAHs. They are purely carbon hydrogen. The second line is carbon hydrogen and at least one nitrogen extra. And then there are many more. What do we do? So this is one example of quinoline, and quinoline you will be meeting again and again in next 10 minutes. This is a molecule. The gray spheres are carbon, the blue one is nitrogen, and the white ones or light gray are hydrogens. This is a molecule, very interesting molecule, and what do? We excite it using extreme UV radiation, and we try to understand how the ionization plus the internal energy of the molecule leads to various types of dissociation mechanism. So this is the binding energy, that means the kind of energy which from where you have removed the electron, so the rest of the energy is left in the molecule, and what all dissociative processes it undergoes. One of the important problem which we tackled with this was whether this molecule produces hydrogen cyanide or hydrogen isocyanide, because this molecule has been detected in meteorite samples. So that means this is there in space. So we want to understand whether it is one of the possible sources of hydrogen cyanide and hydrogen isocyanide. You can always ask why only this molecule. I assure you, whatever you see in this molecule, it is applicable to every PANH which will have similar structure. So whatever is applicable here, it is applicable to all other derivatives of it. So we wanted to check whether it produces hydrogen cyanide or hydrogen isocyanide. We did a lot of interesting quantum structure calculations, and we demonstrated, I'm not going into the details to save time, that this molecule and molecules like this are excellent sources of hydrogen cyanide but not hydrogen isocyanide. Then comes this molecule where the nitrogen has a slightly different location and a different property, and this kind of molecules will produce hydrogen isocyanide. It will dominantly produce hydrogen isocyanide and very, very little hydrogen cyanide, if at all. Again, we did a lot of interesting quantum structure calculations. We also did experiments with extreme UV, and we could conclusively demonstrate this. What is even more interesting is another very puzzling molecule for many astronomers is hydrogen cyanide with hydrogen extra. So this one is another very interesting species because it is actually a radical. It's not stable, but it is seen in many, many areas, especially places like cometary tail, or even in Titan ionosphere this molecule is profusely found. So we found a very nice source for this molecule also. The same quinoline, if you doubly ionize, it produces this. That also we have demonstrated. In this whole evolution, we found that hydrogen migration is a very important step, and it's the same step which actually triggers the isomerization process, and sometimes it ends up producing molecular hydrogen. With our setup at IIST, we could do the first ever experiment where we could explicitly show the elimination of molecular hydrogen experimentally, because it's very difficult to prove that you are eliminating a hydrogen molecule or you are eliminating two hydrogens. So we could explicitly show that result. I am not showing here, that was done few years back using this instrument. So this is the energy correlated time of flight mass spectrometer. So I am just going to now talk about this reaction, this mechanism for next 5 minutes. So we have seen how a simple molecule we take, we break, and we understand the energetics of this breaking process. The same experiment we continued, and purely stroke of luck, we discovered something very, very strange in our instrument. So how the experiment was done, we put a nanosecond ultraviolet laser here. The sample molecule is evaporated in this point, and these green plates what you see are basically plates where we apply electric field. The ions start running in this direction, and using electrostatic analyzer we actually do the energy analysis of these molecules, molecular ions. Now this is again our old fellow quinoline, and its parent mass is 129, so you should have got a peak here, and usually in a mass spectrometer you either get the parent peak or its fragments. That's logical because it will break. What we discover is that we don't get smaller fragments, we get larger fragments. So we started investigating this. Our friend Professor Aravind from IIT Madras had recently published a Nature paper there. He also found something very interesting in similar experiments. So we had a lot of discussion, and then we discovered a very important reason for this kind of thing to happen is that inadvertently the molecules were dimerizing, they were forming a dimer. So now that is not surprising. What is surprising though is normally in a molecular physics experiment, if you want to create dimers of such molecules, you have to spend a lot of money. You really need a very special arrangement to be built to be able to produce these dimers. But these molecules, they simply form dimer. We don't do anything special, we simply evaporate the material at certain temperature, 20 degrees, 30 degrees, they don't have to heat too much. So it's very surprising that these molecules have a tendency to form a dimer just in the evaporative process, and in a minute I will come to why it is important for astronomers. And then in a span of 2 months, my students really worked hard. We worked 24 hours for 2 months. 24 hours we were working, and we discovered 10 molecules, and some more are now. At least 10 already we have discovered where they have this tendency. So again there is some universality, that also we have discovered something but not published yet, that these molecules tend to form dimers very easily simply by evaporative process. Okay, so I'll directly go to the astronomical part. Now I'll quickly digress. In a protoplanetary disk, if you ask anybody which type of reactions you are looking for, the immediate answer would be the reactions on ices or dust surfaces. Why? Because if you are interested in unimolecular dissociation, it's very easy. A single molecule standing in vacuum can absorb energy and break. But a reaction involving two molecules is very complicated, because not only that you have to absorb energy, you have to meet the other molecule. And the easiest way to meet the other molecule is you are sitting on a solid at very low temperature. So the molecules are condensed there, you will meet them. So that's why a large number of experiments are being done on ices and dust grains. But gas phase are usually sidelined, saying it's too improbable. Now here we have a situation where we have proven nitrogenated molecules, and we are demonstrating they have a universal property of simple evaporative process produces dimers, and then these dimers can interact with high energy radiation and produce ion molecule reactions. So we already have demonstrated this using ultraviolet laser multiphoton. Just today morning we finished our experiment with protons. Just now, half an hour back, we stopped our experiment in IUSC New Delhi, where we have tried to show the same thing happens with simply proton collision in the same energy range what the solar radiation produces. Then in June 2025 we are going to conduct 2 experiments, one using a cryogenic ion storage ring in Sweden and another one using a high energy radiation facility in France, where we will do the reverse. If we can produce these ion molecule reactions in one way, we want to demonstrate that the other way is also possible. And there is a reason why that is important. That will be my last slide after this. And then in later this year, we plan to do some more experiments on explicitly selecting the dimers of these molecules and actually conducting experiments using extreme UV, because right now what we are doing in IIST is purely incidental. We are also now focusing more on our Titan reactor, trying to make it better. So the main reason why these studies are now becoming important is because if this is so that it is possible to have such kind of ion molecule reactions, then the obvious question comes to mind is that if you want to propagate a top-down approach, you want to propagate a dissociative or ion molecule reaction channels for the evolution, where did the top come from? And the answer to that is what we are now targeting using these experiments. Again, if you give me another half an hour, I will explain to you how, but don't worry, I will not do that. So not only we are trying to answer how the top to down approach works, we also have to demonstrate how the top actually came to being. Like in case of PHS, it is actually a shock wave which is supposed to process carbonaceous material to produce PAHs. Something has to happen, and something very interesting we are proposing, which if the Caen experiment goes well, we will be able to prove to you. Last 5 minutes I will spend on something else. Our country is very, very well known for having capabilities, but very poor in having the capacity. We have great individuals doing great science, but the capacity doesn't come from individual, capacity comes from organization, and we don't have that. And that's a big problem, and that's why we end up having very big research outputs from individuals or individual groups, and once the group dies, the output dies. So we thought simply doing such very interesting individual experiments is not enough, because we do have enough capability in the country to do astrochemistry, laboratory astrochemistry or laboratory astrophysics. The problem is we don't have a capacity, and that capacity will not come unless we come together and pursue a common goal. And for that we started an initiative called Organics in Space. This is a pan-national, pan-India goal, and some of our friends are here who are part of this initiative. These are all people spread all over India. We are informally involved right now, and we are looking for a formal structure. Hopefully we will have soon one at hand. So the idea is to bring all our capable scientific community together, offer a tangible but challenging problem with global implications, and then use the rapidly growing capacity of our space program. And if you take these 3 things and put together, there are very few things which come out as possible directions. And perhaps the most powerful one is astrophysics, astrochemistry for molecules, trying to identify how the life originated. So this is the main aim of this initiative. This initiative will not only bring the space technology, science, instrumentation, astrochemistry, molecular astrophysics, and contemporary complementary lab experiments, everything together to feed into this kind of research domain. So with this idea in mind, in February 2023, we started one discussion which was part of a national conference. We dedicated half a day session, and we invited even people from ISRO fraternity to join in, and it was a big success, and the idea was taken very well by the community. So we formally launched Organics in Space. The first dedicated symposium for organics in space happened in IIST last year, and it was again attended by 50 faculty scientists, which were only by invitation because it was our first experiment. We were not sure if people will show up, but we really had to stop people. So we had it only 50 and rest were online. So the second one, I am very happy to announce, the second one, our friend Shubhadeep from Geetam University has taken the responsibility, and it will be in the middle of June. These are tentative dates. We are still working with their organization, and we will be coming to ASI also for support, and then this title will change, hopefully. So the idea is to have the second symposium. This will not be by invitation, it will be a normal conference, and this is what we plan to do. This is our website for Organics in Space, and I request you to just join. There is no fees, nothing. This is purely informal right now. There is no official banner yet, but very soon we hope to have one. This is our website. These are our email addresses. If you have any questions, any suggestions, any corrections, please join us and help us. These are my collaborators. I thank all of them. Sarita Vig happens to be collaborator in many ways, including my wife, so she helps. My great friend Dr. C.P. Safwan from IUAC and his team, then the team from outside India. I come from an academic institute, and the most important output from an academic institute is not the papers, not the patents, but the students. So I'm very proud of this slide. These are my past and present PhD students. These are all project staffs who were working with me. These are the students. This is just a partial list of the students who have passed through our lab and now spread all across the world doing science in different areas and technology also. But of course, we haven't done enough. We need to do more. So सितारों के आगे जहाँ और भी है, अभी इसके इम्तिहान और भी है. Thank you.
Thanks for your nice talk, and it's really fascinating to see how experiment can help us to discover space. Now, time for a couple of quick questions.
Thank you so much for this wonderful talk.
Thank you.
So I have a few questions. We talk about HCl and HLC. We see from the conventional chemistry, HCl, HLC is quite an unstable isomer of HCl.
Yes.
So what's the force that's keeping it not to thermodynamically convert into HCl?
A fantastic question, and it is exactly this question which has driven people to this level to really understand. And the only answer which can come to this question is, because you can't magically make it stable in space and not stable in the lab. So the only answer to that is it has to be produced dynamically. Now, what I didn't show is, if you want to produce it dynamically by the bottom-up approach, the number of steps you have to go through and the number of probabilities and reactions you have to deal with is so high. And finally, in all the bottom-up models, by hand some electron density has to be added to make sure that hydrogen isocyanide is produced from bottom up. But top-down approach, we can show that one photon interaction with one molecule you get, and the cometary tail is the one which is dominated by hydrogen isocyanide, not the main body. So it is very clear that you are getting it by a photodissociation process. So it is not stable, it is still unstable. If it meets a surface, it will immediately switch to something else or hydrogen cyanide, but yes, it is dynamically produced in space. And that's why we need to search for mechanisms to produce it dynamically.
So we can go through like from bottom-up approach, we can produce a less stable isomers also?
Yes, it is possible, but the problem is that is a model. Of course, we don't know what is really happening over there, but that's a model. And the problem is there are lots of unknowns which had to be put by hand for that model to successfully produce hydrogen isocyanide. Of course, we cannot claim that it cannot produce hydrogen isocyanide, but I have a simpler mechanism, right, which I can actually show you in the lab. So
Another, I'm like curious, what temperature pressure conditions you maintain while simulating, like what part of it is still the medium you are trying to move?
at present, unfortunately, we don't have the facilities to maintain the temperature of ISM or the outer parts of protoplanetary disk. But that's why we are going to do 2 experiments in Sweden and France, because we don't have that facility here, where we will go down to 10 Kelvin. And all these reactions we will study at 10 Kelvin.
Liton.
So I'm assuming that you also need a certain flux of XUV photons. It's not just that you can get away with 1 or 2, right? So what are the possible environments where you can have this kind of reaction?
Excellent.
The second question is there a time in the history of the formation of the universe and its evolution you think that this kind of reactions would have actually initiated and most of the stuff would have produced larger molecules?
The second one I would answer first. The second one, that is happening even now. If you take what happens to a and now coming to the first one.
I mean the first production of this molecules.
First production, that I don't know. That I don't know. But what we can.
Stars, right?
Huh?
Nucleic stars.
Yes. So okay, then let me go to this picture. This is a picture taken by Alma. Where did it go? Here. This is a picture top right. It's a Alma image, which is mapping different substances. And there people have already detected hydrogen cyanide, hydrogen isocyanide in protoplanetary disk. So clearly at that state, such molecules are formed. Whether it is directly related to these molecules or not, I cannot comment on, because we are right now focusing more on what is happening in our solar system, because that is something we can poke ISRO to, go and check whether it is there or not. So if we build a case there stronger, we will get more resources to go over there. But I believe maybe others have better answer than me. This at a protoplanetary disk itself, this thing should have started. That is clear. But exactly when in the beginning, very hard to tell. Now quickly about the flux of UV or XUV, that is why I showed this picture. The cross section here is so high, and this is for photons. I didn't even show for protons, cosmic rays. It will be even higher. So, and we have shown, which I didn't discuss, that once you excite this plasmon, these reactions start. Explicitly we have shown this. Only one such set of such experiments I have ever done, where we have explicitly demonstrated that you excite a plasmon and this kind of isomerization, dissociation, these reactions start. And they are quantified also by us. So we know that this happens. So you don't really require very high flux, very small flux, cross sections are high, this will happen. Yeah. Thank you.
So I am interested in the formation of dimers. So you know that quinolene, for example, can form dimers. And so is it possible for normal pHs also to see such kind of dimers?
Excellent.
The other thing is that can you predict about how these dimers are arranged, for example, parallel configuration or T kind of configuration? The third thing is, can we do spectroscopy? So I am a pH person, so I have got
Excellent. Yes. Okay, to be very frank, a lot of these questions are haunting us also at present. Where is my list of? A lot of these questions are haunting us also right now, because it's just 6 months back we sort of started discovering that this is something universal. Okay, we are not the first ones to discover this. Arvind's group found it almost 2 years back, but only in one molecule, and the mechanism which they predicted is most likely not correct. They didn't explain it by the basis of dimerization, another process. But we are explicitly proving that it is a dimer or a trimer which is producing this. First. Second, pHs can also produce dimers, and people have studied them. But since these reactions themselves were never focus of those studies, the focus remained on finding out whether a dimer or trimer is produced or not. So now there is a need to repeat those experiments and check whether there was a ion molecule reaction possible.
So is it possible in your lab for normal reactions without nitrogen?
So that is where our limitation comes. For us it was incidental. Why it was incidental is because whenever we discovered this, whenever nitrogen is sitting inside the ring or it is sitting with an sp2 hybridization, this dimerization is strong enough that pure evaporative condition you will get at room temperature, you will get dimers. But whether that is because for producing dimers of naphthalene and anthracene, people really had to do build special apparatus for that. We don't have that. So at present we cannot do. But we have collaborators and they are interested now to take it up. So we will look for it. But it's just that will require vibrational spectroscopy, that even my friends right now do not have the capability. But if we manage to do this experiment with an ion storage ring in Sweden, in principle, because there the temperatures are really 10 Kelvin and it is stored for seconds. So we have a laser there which is deep infrared, we can actually find out whether it is its configuration, but right now that is not the focus. So, but yes, it is very important problem, I know.
Okay, for the sake of
Thank
time, I think let's thank again Umesh for
Thank you.
the talk. Let's welcome Professor Sudha Rajamani for our next talk. She'll be talking about the astrobiological narrative of light's origin on the pale blue dot. So it will be exciting talk. Sudha, I will.
Let's see if you can find them. It has to go.
Just over your ears.
Yeah. Where's the mic? there. Yeah. अच्छा ये भी कान के ऊपर जाएगा?
Yeah.
Okay. Hang on 1 second. I have hair. What can I do? Hair, chashma. Okay, that's okay. I think that'll stay. This is forward, this is back, and that's the pointer. Good morning, everyone. Can you hear me? Yeah. Thank you so much to ASI 25 team for having me over. Particularly thanks to Jasjeet, because of whom I'm here. I've been wanting to come to ASI for a while, but I wasn't sure if I would be a fit. I still feel very much like a fish out of water because I'm the only one here with my kind of background, which is biochemistry and biology. Having said that, this is something that happened very commonly while I was sort of postdocing in the US, where we would meet people via a triennial meeting. This is the International Astronomical Union Commission 61 or something. I forget the precise details, but so it's very nice to come back and listen to all these very interesting talks. Umesh has set the stage wonderfully for what I'm going to be sort of taking off from. Having said that, Umesh, I have a gripe towards the end. You just kept harping about astrophysics and astrochemistry, and you left astrobiology out, especially in light of Ryugu results. Astrobiology is still very, very right there with astrophysics and astrochemistry. And I'm hoping that by the end of this talk, I'll give you some flavor of how we in the field think about the transition of non-life to life that would have happened on the early Earth. And I'm talking about events that panned out possibly 4 billion years ago. So hang in there, and I'm not going to be talking about the research that happens in my lab per se. I'll give you some flavor of it, but the goal is to give you a much more broader overview of how these questions are answered in the field. Everybody here knows about the pale blue dot, so I'm going to keep it very short and say that this played a very important, this picture taken in 1990, by Voyager 1, actually inspired Carl Sagan to title his book, The Pale Blue Dot, A Vision of Human Future in Space. And today, a couple of decades down the line, it very much is going to be a reality very, very soon. And the one thing I want you to keep in mind is this pale blue dot that you see here. Let me find the arrow, yeah. I've circled it. I don't know how much you people can see from the back there. A very important thing to remember. Look at it again. This is what he said. Look again at that dot. That's here, that's home, that's us. We do not know of a single other entity, either in our solar system or beyond, that hosts life as we understand and appreciate it. And I will give you today a sense of how we think this emerged on the early Earth. Astrobiology is a field of study that deals with understanding the origins, the evolution, and distribution of life in the universe. Inherently, it's an extremely interdisciplinary subject. What I've mentioned here is not encompassing, but it feeds off of very diverse areas of sciences. And this is a question that has been, plaguing humankind for time immemorial. Understanding, are we alone in the universe? How did we even come about in the first place? And is there life elsewhere out there? And this has been a significant motivator for card-carrying astrobiologists like me to actually structure our questions in a systematic manner and actually try and understand, both using experiments and modeling approaches, to appreciate what kind of environments, niches, and processes would have driven this transition. And given that it is such an interdisciplinary area of sciences, it is of immense value to drive STEM education and excite younger generation about taking on STEM careers for their future. So doing outreach in astrobiology, astrochemistry, super crucial. Some of the key topics that people in the field deal with involve understanding the origins of life. Did it happen on terra firma? Did it come from outer space? How did the transition happen, whatever the source was? Understanding the habitability and limits of life, understanding life that lives at extremes, also characterizing extreme environments that can host life in those environments. How do organisms survive and adapt? Because all of this has fundamental implications for where you can look for life. And when you talk about looking for life, one thing is super crucial. You need to understand the signatures of life. So there's a community in the field that tries to characterize them systematically, tries to understand biomineralization and fossilization processes, how do biomolecules come about, what kind of transformations they undergo, and what does that mean for biosignature characterization for life detection purposes. So it's sort of a very broad area of set of questions that we look at. Today I'm going to be focusing on understanding the origins of life. There are a few different hypotheses prevalent in the field, but I'm going to focus on one of the most prevalent one because it has more proof than anything else. Before we go on and talk about how it came about, life came about, you need to understand that we need to have a working definition. You go Google the definition of life, there's going to be answers that get thrown up. So NASA Astrobiology Institute constituted a working group comprising of the scientists from diverse areas, including astrochemists, and all the way from astrochemistry to molecular biology, and they came up with this working definition. Life is a self-sustaining system, chemical system, which is capable of Darwinian evolution. So if you can concoct an entity that can encapsulate all of these three important hallmarks of life that are mentioned here, which is the capacity to self-replicate. Life is encoded in information, and that information needs to be propagated in order for it to survive. There is an important need for some sort of a metabolic process that allows it to survive far from equilibrium, because equilibrium means death. And importantly, all of these very interesting lifelike reactions are happening in constrained spaces where compartmentalization plays a very important role. And I'll be touching upon some of these as we go along. So if you can concoct a minimal entity using a bottom-up approach, he kept talking about top-down. I'm going to be talking about bottom-up, but from his top-down place. Yeah. So if you think about ways to bring molecules together and concoct a living entity in the lab, it means you have been able to crack the code of how life came about on Earth. Just four important events to set the stage, to sort of give the, we need this background in order to be talking about other things further down. The formation of the solar system would have played a very crucial role because our planet wouldn't have been present otherwise. This is thought to have happened about 4.57 billion years ago, and soon thereafter, Moon formed by the giant impact between Earth and a Mars-sized object called Theia, and Moon's presence has fundamental implications for a lot of environmental constraints that we work with when we are trying to simulate early Earth conditions. This is a very important but very debated sort of proposition, but Moon has signatures of this. Very early on in Earth's history, it is thought that there was constant bombardment by extraterrestrial objects, meteorites, comets, asteroids, and this violent period was not just violent, it was super crucial because it brought in a lot of interesting organics that Umesh was talking about that is formed in the interstellar medium. This is still debated. How did the Earth's crust form? Or not how, when did the Earth's crust form, and when did all of the water vapor that got carried, or the ice crystals that got carried via comets and meteorites, condense to form water, which is super crucial to life. No water, no life. Water is the canvas on which life reactions are possible. So I'm going to be talking a lot about aqueous reactions, not as much as gas phase reactions. But then how do you think about this question? This transition from non-life to life becomes immediately apparent when you look at this beautiful continuum that exists between physics, chemistry, and biology. Down here is biological molecules. I've just shown two of them. One is a proteinaceous enzyme and one is an RNA enzyme. Zoom into them. They're made up of fundamentally monomeric molecules which are strung together via covalent linkages. Zoom further into them. What you see is they are made up of chemical entities, some of which are very readily formed in the interstellar medium. Further zooming into them essentially shows you that there are a bunch of atoms that are held together to form simple molecules. So given this continuum, the question that we are asking about transition from non-life to life can be structured around the fundamental understanding of how did chemistry transition to biology on the early Earth. And when we talk about chemistry, the first thing to ask is, if you're talking about transitions of molecules, simple molecules to more complex biological molecules, where did those simpler monomeric entities come from? There are a few different hypotheses, but two prominent ones involve synthesis that happen on Earth's on terra firma in certain environments where very interesting fluxes could be facilitated, temperature fluxes, chemical fluxes, so on and so forth. And that is shown here as a artistic rendition of a terrestrial geothermal environment. A lot of interesting action in terms of gases, in terms of molecules that can form that result from the rock-water interactions, and the day-night cycles can lead to a lot of interesting fluxes that can be facilitated in these conditions. The other important source is that late heavy bombardment period that I talked to you about, probably brought in a lot of very relevant organics. And again, remotely not encompassing of what the list of prebiotic inventory of molecules would be, but these fundamental biomolecules could have been readily plausible on the early Earth. So given their availability, how did they go from being simple molecules to much more complex molecules? So if you look at what we are made of, if you take the cells from our body and open it and study the components, we actually are made up of four important biomolecules. Nucleic acids, which encode information, this is your DNA and RNA. Proteins, which facilitate reactions. Carbohydrates, which are sort of pin codes for cells and play a very important role in some higher order functions, so we can keep them aside. And the fourth important class of molecules, any guesses? Loud? What are cells made of? A membrane. The membrane is essentially a lipidic entity. Lipids are amphiphilic molecules. When you take a bunch of them, put them in water, simple thermodynamics will drive them into forming balls where all the water-hating molecules go inside and the water-loving molecules stick outside. This is just your soap formation, right? Exactly like that. So when we think about understanding origins of primordial life, three molecules are sufficient to talk about the processes: your nucleic acid, your proteinaceous entity, and your lipidic compartmentalization entity. I'm not touching upon certain other processes, but there's a lot of interesting atmospheric synthesis that would have also added to the inventory, along with hydrothermal processes which happen on the ocean floor, and other interesting interfacial chemistry which could have also made the prebiotic soup a very, very rich soup of heterogeneous molecules, out of which only a subset of them got selected for life over the billions of years that happened, or several hundred millions of years before primordial life came about. This is just quickly to show that extraterrestrial exogenous delivery, I'm going to call it exogenous delivery, was very readily possible. And here is the famous example of the Murchison meteorite, which fell in 1969 in Victoria in Australia. You can't see it, I'm sorry, but it is comprised of all the important molecules that you can, many of the important molecules that you see in life today. Amino acids, aliphatic hydrocarbons, aromatic hydrocarbons, carboxylic acids, purines and pyrimidines, which are the informational carrying moiety in your nucleic acids. And much more recently, as Umesh already alluded to, Ryugu, this small 5.1 gram material that was brought back by Hayabusa 2, basically showed us that some of the more complex molecules, more complex than the astrochemical molecules, are readily formed in the interstellar medium. The why and how is for him to figure out. I'm going to talk more about how do you go from them to molecules of life. And this is an artistic rendition just saying that these kinds of environments would have been where, even though they look extremely inhospitable, this is where life reactions are thought to have come about. And given the right set of ingredients in that mix, you actually can have very interesting molecular interactions that can lead to emergence and evolution of molecules. And the possibilities are endless in principle, because there's a whole bunch of molecules present in the prebiotic soup. And one of the problems we have in astrobiology is to constrain and try and understand how do you go from this complex, smaller molecular space to more sort of fine-tuned, higher order molecules which got selected for due to probably various environmental conditions. So the abiogenic theory proposes the following. Simple molecules which were present form the precursor molecules. The precursor molecules come together to form the building blocks of life, which then undergo self-aggregation or oligomerization to form some of the important molecules that we already talked about to result in primordial life or cells. All of life is cellular in nature excepting for viruses, and by definition that I talked to you about, virus is not alive. So we can think about now the problem as how do you go from a soup of molecules to the oldest cells that might have populated the earth 3.8 to 4 billion years ago. And here is the timeline, rough timeline when these events are thought to have happened. 4 to 4.2 billion years ago itself, right soon after the formation of earth, interesting chemistry was going on. How did you go from there to what is called an RNA world? RNA world is a very interesting hypothesis in the field which posits that very early life would have used only one main molecule to both encode information and catalyze reactions. And RNA is this one super awesome molecule. Unlike DNA, its cousin, which is very, very limited in what it can do, RNA is extremely versatile. So it's thought that very early on in life, primordial organisms probably just used an RNA-based life process. And two main environments that would have supported these reactions are both volcanic in nature. One exists on terra firma and one exists on the ocean floor. I'm going to limit myself to this environment. I'm going to be talking about the terrestrial hypothesis of origins of life, and I've touched upon a lot of the steps already. And what we then ask is how do you go from the prebiotic soup of molecules to what is the least common denominator of all life that we know today, all the way from single cell organisms to the most complex humans. Those are cells. So a whole bunch of steps exist and that you can think of, and where we work in our own lab in all these areas. You have to think about the prebiotic chemistry and the formation of the building blocks. Then you have to think about how they oligomerized. He stopped with dimerization. Biology can't just work with dimers. It uses dimers for certain important processes, but it requires much, much longer polymers. So how did these polymerization or oligomerization processes come about? And how did they effectively result in functional molecules, even in the absence of highly engineered machines called proteins? Once interesting molecules came about, how did the information get propagated? Kind of crosstalks happen between the minimal enzymes and the compartment molecules, the lipid molecules. When we're thinking about how do you go from a bag of chemical molecules to a cell comprised, so to speak. So I want to finish this part by saying that a recipe for life itself was readily plausible. From the same precursor molecules, inorganic molecules, you can make the fundamental precursors for making amino acids, for making nucleosides which form nucleic acids, and for making molecules that would have led to the formation of membrane molecules. So we can now systematically address some of these questions in the lab by simulating early Earth conditions and asking, how do you go from monomers to oligomers, and how can you understand these cell-like entities that you make in the test tube, and how can you show that they can embody life-like properties of growth, replication, and evolution. What did the earliest life forms look like? We don't know. The primordial cell or the proto-cell, probably small, tennis ball, you cut open it, outer is the membrane, inner you have an encapsulated genetic material and potentially a small network of molecules that allow for simple metabolism. But what we understand and appreciate in biology already are these earliest forms of life called prokaryotes. Some of the oldest signatures are from 3.8 billion years ago. 3.8 billion years ago, which is only a couple of hundreds of million years soon after the Earth formed. So whatever happened, whatever kick-started primordial life, these reactions happened very, very quickly soon after the formation of Earth, possibly in certain niches which were more protected than the ones which were exposed to very harsh UV radiation and other kinds of denaturing sort of conditions. And here is that evidence. These pillow-like, stone-like structures are called stromatolites, and these stromatolites, both dead and living stromatolites, are found in Australia on the western coast. They are essentially a product of biology and geology, and you can take them and study them to date how old they are. And some of these have been dated strongly to 3.5 billion years ago, and the biological component comes from cyanobacteria, a very important prokaryote, which emerged from that primordial community of cells. And please also remember that all of those reactions happened completely under anoxic conditions. No oxygen back then, yeah, very early on. If you put it on a 24-hour clock, roughly by noon, you had these prokaryotic fossils come about. Can anybody tell me what is the important thing that these prokaryotes did to our planet, which changed the planet's naksha, which changed the planet's atmosphere, which completely changed the planet's capability? Any guesses? Oxygenation. Somebody said it. I can't hear loud enough. You should have been louder. Essentially, they used solar energy. They took water and carbon dioxide from the medium. They cooked food that fueled the metabolism, and a byproduct was oxygen, which got spit out. And all of the oxygen that you see in the atmosphere today is thought to have come from this process. Yes, it would have been poisonous for some of the early organisms, but some of them did learn to adapt and survive and evolve. And these primordial entities, if you can appreciate it, this rendition basically shows they themselves are extremely complex. Here are cells that you and I are made of. We are all eukaryotes. We are much more higher ordered organisms because we have a lot of membrane-bound organelles in our system. But these guys, which are very, very relatively simpler, they can do something amazing. They can exist in extreme conditions and thrive where you and I will die out. High UV conditions, high salt conditions, extremely low freezing conditions. Name it, you can find life entities living there. So it's also a very important approach that people in the field use to understand the limits of life. If you're looking for life outside of our Earth, within our solar system or beyond, the conditions are pretty extreme. So it's important to also understand the biology of these organisms and how they manage to survive and adapt to better be able to design our missions effectively to land at the perfect location, to sort of look for the right kind of molecules, so on and so forth. And when did we come about? Our ancestors came about 2 billion years ago, which is equivalent to afternoon on this 24-hour clock. So when did you and I come about? Ultra close to midnight, just ultra recently, yeah? And look at what we've managed to do to our pale blue dot already. So the youngsters in this room, I'm really sorry that we already screwed it up for you, but the onus is on all of us now to try and remedy, because building habitats outside of our Earth, even though is a very important way of thinking about, how can we escape from a world that will not be sustainable any longer, it's also very important to understand that we do not know of any other body that can host life as we understand it. So it's super crucial to protect whatever we have. So I'm hoping that you take away from this that what you can do, the best of a story that you can draw upon, is what you get consistently from all the evidence. The biological evidence, the geological evidence, the chemical evidence, all of it allows us to put together the pieces of this puzzle, which eventually is bringing to view what might have happened potentially. We will never know if this is how it happened, but there's reason to believe that it possibly happened this way because of the propensity with which these reactions are readily plausible. We not only, the other very important thing in astrobiology research is you cannot be doing only highly controlled laboratory experiments to understand what you're interested in. You need to go out into analog environments to understand whether the reactions that you are looking at will hold good in more complex environments. And for this, we go out to what are called analog environments, and we have a very important Mars analog on Earth. We have a few different Mars analogs on Earth, and one of them, I'm sorry, I think that slide is not here, so I'll just go back. But a lot of this study has led to very interesting, outcomes, and you have Perseverance right now sitting on Mars trying to understand whether at some point in its very early history, did it host life? The outer crust, the outer regions of Mars completely dead, but the subsurface could possibly have held water. So could you have had life in those environments is a question which we are very interested in understanding. So this brings me to a very important last point I'll discuss in a few slides. All of the appreciation that we are gleaning from these experiments that we do is also helping us to set the stage for asking certain very important questions. If we can delineate the path that happen on Earth where chemistry transitioned to biology, can you take this understanding and ask a very, very crucial question, is there life elsewhere in the universe? So if you have the just right planets in the habitable zones around the stars that around their parent stars, can you actually try and look for signatures that will tell us if they are on the way to hosting life, or if they host life, or if they did host life at some point in their distant past? And these zones are called the Goldilocks zones, where the conditions might be just right, neither too hot nor too cold for life, just like it was on Earth even 4 billion years ago. And the planet needs to have a suitable atmosphere. And this is where we rely heavily on exoplanet researchers to tell us the environments about the environments that are on these planets, so that we can better think about whether that might be suitable for hosting life. But please appreciate that signs of habitability is not equivalent to signs of life, because a planet is basically a product of a lot of important processes. A closed system will only account for the atmospheric model, where its atmosphere is a closed sort of an entity, but that's not true. Atmosphere is anything but closed, because there's a lot of exogenic processes that will alter the atmosphere, impacts that impact processes, light escape, hydrogen and helium escape, so on and so forth. And a lot of interesting geological activity, seismic activity that happens can temporarily change the composition of a planet. Now put biology on top of already these complex processes, and you are going to have a very, very complicated picture which will start emerging, and then then it begs the question, how many of the planets or within our solar system or other moons of planets within our solar system or exoplanets can actually have all of this come together just in the right sort of proportions for things to have happened? We do not know, but we can look for them in the form of biosignatures, which are essentially patterns, substances, molecules, or ensembles of all of this that will indicate whether it is hosting life or it hosted life in the distant past. And they have to be global for better. These biosignatures have to be on a global scale to enable detection, because it has fundamental implications for the modification that they bring about on a planet. They change the planet's atmosphere, its surface, its appearance over time, so much so that you will not appreciate the fact that Earth probably looked like this when it first formed. Now it's this beautiful pale blue dot, but it wasn't like this when it first formed. And they should always, biosignatures should always be assessed in the context of the planet's environment. Without that, you will never be able to understand whether life could have been possible. And I'm going to finish off by saying that you have a whole bunch of detection methods, and this is something that the astronomy community is very familiar with, but it allows you to look at substances all the way from angstrom to several kilometer ranges and understand these things. So this is what I was referring to as planetary analog studies, where we go out to environments that look like ancient or current Mars. In this case, it's Ladakh, and we try to understand how things would pan out in realistic conditions, and it has implications for geomorphological, astrobiological research, and also for instrument testing when you want to send things out. And with that, I think I'm done. We are very interested in understanding if there is the possibility for habitability in other bodies in our own solar system, and some of the important candidates are Mars, Europa, and Enceladus for a lot of the important prerequisites that I've already touched upon earlier in the talk. I thank you for your time. I'm happy to take questions.
Thank you, Sudha. Time for questions.
How is habitable zone? My question relates to the evolution part of life. We normally, the evolution is driven by mutation, which is supposed to happen randomly, intrinsically in the addiction process. My question is about the importance of radiation and cosmic rays. Do we understand which process is dominant, intrinsic random mutation or cosmic ray induced mutation? Because that will affect which planets you are looking for. If the planet is exposed to more radiation, is it possible evolution has a more faster rate of?
So there are two ways to think about this problem. One is very damaging radiation, which basically will kill whatever molecules are formed in the first place. That is one way. But if it's not extreme, and if it is more in the regime where it's leading to a lot of mutations where things are happening, you're actually posing a question which we think about a lot, and I will restate your question. Whatever we appreciate about the transitions that happen on Earth and the biochemistry that it has led to, is it what we are supposed to be looking for when we're looking for life elsewhere in the universe? If you look at just Earth, all of life that you see on Earth is made up of the same set of molecules, CHNOPS. And the CHNOPS set of atoms came together in very interesting myriad ways, and that was shaped by the planetary environment that it was happening in, which eventually led to this. In the lab, I can actually make informational molecules which look a lot like your RNA and DNA, but are not made up of the exact same chemicals. And I can still show you that they are informational coding molecules. They can propagate information, and are they possibly those molecules that we should be looking for elsewhere? Hard to say, which is why there's a big debate going on in the field. Some people in the biosignature field have gone to a point where they say biosignatures are really bogus. We should be thinking about something else in a more systematic manner. I'm going to be one of those very few people that argues that you have to look for prebiotic signatures, because a lot of the chemistry that we do in the lab, prebiotic chemistry that we do in the lab, has humbled us to a point where we know that what we see is not what we get. We get a host of other molecules and we ignore them. We ignore them because we come wearing these blinders and we appreciate only what is present in our biology. So the way to answer very tangentially your question is, we should be looking for a whole suite of molecules, all of which might not be hardcore biosignatures, but still give you an indication for whether that particular system is on its way to transitioning. And there's enough reason to believe that I can come up with a catalog of those molecules.
You are in favor of or working towards the terrestrial origin of the life. So how you explain like the prebiotic soup or the Steller-Miller synthesis of amino acids, this all are on the Earth, within the Earth atmosphere, that's what you work on?
The terrestrial origins of life posits that given the right environment with the right ingredients, the transition from chemistry to biology is possible. However, the source could have also been exogenous. It doesn't only have to be terrestrial, which is why I kept talking to you even about exogenous delivery of molecules. Molecules, not life, of molecules.
Okay, so the meteorite, they have amino acids, almost all amino acids that we have in life.
And more.
And more are in the LIGO form and sugars in the dextro form. So what you comment over like we get from the outer space or
We-
they persist within the Earth?
So here's the thing, and that is something that Umesh touched upon, and my understanding is that you very readily find enantiomeric mixes of molecules in the interstellar medium chemistry. However, you see a preference for a certain handedness in biology. Your nucleic acids are left-handed and your sugars are right-handed. How did this come about is a big question. There is one thought particularly that talks about a runaway process that sort of, even if you have a 1% excess of one over the other, you can have runaway processes that would select for that. And eventually the handedness of one molecule dictated what the handedness of the other molecule has to be. Now all of that I'm saying is very faffy. There is very little research that proves this. That is rooted in hardcore chemistry. When you move to the biomolecular space, and if you're still in the non-enzymatic prebiotic space, there are only a handful of experiments that show that indeed, at least for the selection, you could have had some constraints come about because one molecule already developed a certain kind of handedness, and that forced the other molecule to be opposite for better interaction. Maybe that is what drove it. However, we really do not know what exactly led to this.
Right. Is there anything specific that's required for the life to evolve into an intelligent life, if that's what we call it, or does it naturally happen if the life can sustain for a given number of time?
You're not talking about evolution of cognition. So there are people that think quite a bit about this, and there is reason to believe that if you have enough time to evolve certain processes, and if certain selection pressures forced you to, come up with more exciting solutions which don't randomly happen by chance, there are sort of imperatives that will force the brain to develop much more into a much more complex entity. I do not know too much about how people think about when these transitions happened, but we have some sense of which were the first organisms in which a primordial brain came about. But is there a notion of self in those? We do not know. What we understand about notion of self, and they all are in the higher order mammals. So yeah, maybe given enough time, the chances of this is very high. I think the bigger problem is transitioning from chemistry to biology. Once that happens, other processes probably are inevitable, unless a catastrophe or happens like one of your extinction events and wipes every life as we understand off, resets the clock, and it's only the microbial entity or the very small entities that are alive, and once again they have to evolve. But I think that process is inevitable. The original transition, more difficult.
Yeah, I was intrigued by the meteoritic evidence that you brought, the evidence of organic molecules on the asteroids and on the meteoritic samples that you mentioned. I was wondering whether the detection of the organic molecules is on the surface of those elements?
No.
Whether in the sample that was brought back from the asteroid, what depth was it brought back? One implies that you are looking at material gathered up, swept up from the ISM, while the other implies it was formed in situ while the meteorite or the asteroid was formed. So, which is it?
Both of your possibilities still happened, outside of Earth. There's a third possibility which we think a lot about while we're trying to do some of these studies, is that we don't have cross-contamination from Earth or Earth biochemistry. Having said that, if you make sure that you are drilling to the core of these entities, particularly meteorites, they just fall. And you just go, you retrieve them, and then you work with them. So you then make sure that you literally only drill to the core. And I don't know the exact sizes that people work with, but only that core will enter a clean lab. And once it goes to a clean lab, it undergoes a whole bunch of processes where you're trying to extract the organic phase material, the aqueous phase material, and then you start analyzing it. It was in very fine print there. The Murchison meteorite throws up 100 amino acids. 100. What we have in biology now is 22. So that in itself makes it abundantly clear that rest of it is not possible on terra firma. We haven't seen them yet. So they had to happen somewhere else, which is probably in the interstellar medium.
We haven't seen them yet under lab conditions, which have a certain range of parameters for which they are generated, or we cannot generate them at all under
that question is not answerable because we have not really systematically figured out every single combination of parameters to date. Maybe at some point, like he pointed out, they're going to go to France and they're going to have access to ISM-like conditions and things can be more plausible there. That is the rate limiting step. You need to be able to simulate some of these extreme conditions, and then the question is, can you actually even make them in the lab? And the answer could be a yes, we do not know yet, as yet.
Thank you very much.
Thanks.
Yeah. So my question is, there is a famous hypothesis that tells comets have brought life on Earth. So what about the latest development in this hypothesis?
Yeah, that hypothesis is very problematic. One can always argue that microbial life that got, sequestered in the core can survive. Spore-like conditions possible, I don't know. I have a bigger problem with transferring the problem of understanding the process of this transition from non-life to life from a planet whose history, geological and planetary formation, we are still trying to figure out. वो problem को यहाँ से लेके कहीं और भेज देंगे और उस other outside earth के conditions का किसी को कुछ भी पता नहीं. So I think if we ever get to a point where technologically we get that advanced that we can ask some of these questions, the answer is yes. But I will still say that panspermia hypothesis, where it's posited that life rode and got seeded, if it were that readily plausible, why are we not seeing life elsewhere? Even within our solar system, maybe Venus's terrestrial atmospheres are very, very, extremes in temperatures, but the clouds have some ambient temperatures. Yes, they are acidic in nature, but we have acidophiles on Earth, but we don't see anything there. We have not yet found life on Mars or signatures of it. We only have seen water as of now. I would imagine that if it's panspermia, the chances of being able to record these so-called biosignatures will be very, very high, because also the assumption automatically could reasonably be that, given it's the same set of atoms in the ISM, the chemistries that made them plausible, the chemistries that resulted in molecules could be reasonably similar. No. So exogenous delivery of molecules, totally fine. Exogenous delivery of life, I can't wrap my head around it, and there hasn't been any research beyond the famous Vikramasinghe hypothesis that was posited a long time ago, and it's a hypothesis. They never showed anything.
Yes. Thank you. Yeah. So I think we can continue our discussion during topic break.
Sure.
So, thanks, Sudha, again for your talk. Now, let's welcome Priyanka Chaturvedi. She is going to talk about exoplanet characterizations from Jupiter's to Earth, the way forward.
You got it?
Yes.
Hello? I don't think so.
Can you test now?
Hello?
This is to go next, previous, and this is the pointer.
This is to go next. Previous. Okay. So, very good day, good morning to everyone. It's a great pleasure to be present here at NIT Rourkela, and I would like to thank ASI for giving me this opportunity to present my research work. So, the previous speakers have talked about laboratory astrophysics and also talked about some bits of solar system studies that we are talking about in broad field of exoplanets. I will be taking you right away outside the solar system and talk to you about how we are really detecting exoplanets and how about the characterization. So first I will begin by talking about large planets, the Jupiter-sized planets. This is where the first exoplanets, the kind of first exoplanets were detected. Next, we will, on the journey, talk about the smaller planets. While we are building this perspective, we will also talk about that in the beginning, most of the studies were concentrated around the solar kind of stars, but the paradigm has recently been shifted to look at different kinds of stars around which exoplanets are present or not. So I would be touching some of these themes. And while we are talking about the smallest of exoplanets, there are also some nuances in detecting these systems. So I will be also touching upon these aspects and how do we deal about this. So as I begin my talk, I would like to thank all my collaborators and my mentors and students with whom I worked with in the past decade or so. They have been responsible for the some bit of the work that I will be showing you here. So the funding agencies and the institutes I worked with previously. So thanks a lot to all of them. So let's begin with the first detection of the exoplanet. The first formal beginning of the field happened to be nearly 3 decades ago with the first detection of exoplanet around the star 51 Pegasi b. Though we spoke about this even yesterday in Joe's talk, the first detection of exoplanets was not around a main sequence star, but around the pulsar, a star which is towards the end of stellar evolution. But there haven't been many discoveries around pulsar-like planets. But the field of exoplanets triggered after this first discovery around a main sequence star. These two gentlemen that you see here, Didier Queloz and Michel Mayor. So they won the Nobel Prize in 2019 for discovering a planet. So why they won this Nobel Prize? Up till before this time, what scientists were really looking for were planets in a solar system expecting what we should have in our solar system. So just to bring into context, the nearest planet in our solar system is Mercury, which orbits at 88 days from Sun. But here we found a planet which was half the mass of Jupiter, but it was orbiting its host star at an orbital period of 4.2 days. Having a planet that massive and being so close to its host star was something which was not really thought of before. And this completely changed the way how we looked at planets now. Shortly thereafter, there had been many discoveries which mostly were around this regime of planets, which means the Jupiter-like planets. So we will be slowly talking about this planet detection method in the next slide. Let us look at this particular slide. This is the total number of exoplanets which are discovered by the year-wise discovery. So different detection methods are colored here. What I would like to point your attention to, most of the exoplanets detected are pointed out in this purple color and green color. The purple color method is the method which is the transit detection method, and the green ones are the one which is the radial velocity method. As you can see from this slide, the initial discoveries were mostly by the radial velocity method, which was greatly boosted by the work done by Michel Mayor and colleagues. Then later on, we had exciting transit space-based missions that were launched somewhere around 2009, and which also had another boost when in 2015 and 18, we will be talking about some of these over the course of the talk. So mostly my talk will be related, will be spoken about these two detection methods. And so let us begin with the first one, which is the radial velocity method. I believe most of us understand what the method is, but just to bring into perspective, if you have a star and a planet system, you're not going to take any light from the planet. But because of the presence of the planet, the star does a small wobble around the center of mass. So if you're taking a spectra of the star at each location, then you would be able to detect this wobble if in the detector regime you are having a very high resolution spectrograph. High resolution spectrograph simply means that you are able to resolve the spectral features on the surface of a star at a very high resolution. So two neighboring atmospheric lines can be really separated out very well. So this method works when the star and a planet system is edge-on. So any motion of the star in this plane is something which is able to be recorded. Anything which is face-on this system would not be able to be recorded or would not have any significant measurements. So how does this really work? So let us try to look at this in a more elaborate fashion. So what we have is, you have a spectra, which you see in the dotted lines here, and you cross correlate it against a stellar binary mask. So a stellar binary mask is essentially the exact position where you would expect your lines to be if the star would be stationary. When you're cross correlating all the atmospheric lines against this stationary mass, it is going to build a cross correlation function peak at the location of the delta wavelength shift. So many stellar atmospheric lines are used to create this they are used in this radial velocity method. So naturally the stars which have many atmospheric lines will be the ones which will give good radial velocity precision. Since solar type stars are known to have sufficient atmospheric or photospheric lines on the surface, they serve as excellent candidates to perform radial velocity studies. Hence, these were the stars which were first looked for detecting exoplanets around them by this method. What we do next is, whatever wavelength shift that we have captured or which we have measured by the high resolution spectrograph, we convert this wavelength shift into velocity shift by the simple conversion that your delta lambda by lambda is nothing but your delta v by c. So anything which you saw on the linear axis, the wavelength shift, that gets converted into the velocity. So this is a typical diagram of a radial velocity versus time, because you're taking spectra of the star at several instances while the star is moving across the orbit of the star planet system. Any positions between the two peaks will tell you the period of this planetary system, and the peak or the semi-amplitude will tell you how massive the planet is. To put into context, just look at this equation which lies in the bottom here. So everything is in the units of sun and earth. So if you put in all the numbers here, in order to detect an earth-like planet, the radial velocity semi-amplitude that you need is around 9 centimeters per second. So an Earth-like planet in a sun-like orbit which is separated by 1 AU, that is the typical radial velocity semi-amplitude that we are talking. This is really very small. In order to detect this, it's not very easy. Let us talk about Jupiter-like planets. A Jupiter-like planet, in the same formula, if you plug in the numbers, this comes out to be 12 meters per second. So a Jupiter-like planet separated at 5 AU, which is in our solar system, we would need a radial velocity semi-amplitude of 12 meters per second. So this is certainly more doable and very much doable in the current conditions. To reach 9 centimeters per second is still something which scientists are trying to move into this direction who are working in the field of exoplanets. We also spoke about the transit method, which has been the most successful method. It is very easy to understand. If you're trying to look at the total light which is coming from the star at all the times, the moment when the planet passes in front of the star, you will see a dip in the light curve. So this is responsible for the detection of the exoplanet. So despite the fact that the early on detections in the field of radial velocity methods started in 1995, the first transiting exoplanet was only confirmed in the year 2000. Because doing transits from ground is also not very easy. There is a lot of stuff happening in the exoplanet, in the Earth's atmosphere, which affects this kind of detection. So when you take these detections to space, naturally you are eased out. The other bit is you should also be aware of when the central transit time of the system is occurring. So if you want to plan exoplanet detection studies on a large scale, what ideally one would be doing is looking at a large field of view and trying to take transits of many such candidates and then figuring out if any transit signature is present or not. Now, as we spoke about the first planets that were detected, we spoke that many early on Jupiter-like planets which were close to their host stars were detected. First of all, the transit signatures were very prominent, also because of their masses and the radii. They were also situated close to the host star. They produced large semi-amplitudes of the orders of 50 to 100 meters per second. So they were very easy to detect with early technologies which were developed to detect exoplanets. Now some of these planets, since they had masses which was equivalent to that of Jupiter masses, but they were orbiting at a period less than 10 days, the term hot Jupiters was coined. So this was to identify a separate class of planets because they were completely distinct of Jupiter-sized planets that we see in our solar system, the Jupiter that we see in our solar system. It's very interesting to think about such objects, because what we understand from our solar system formation, that in order to form a Jupiter, you need some solid material which should be present at the snow line, from where you should expect the formation of Jupiter. But in this context, you have something which is placed very close to its host star. How these planets would be forming? Do they form ex situ, which means they form at a very greater distance and then the disk disappears, and because of some eccentricity excitation, tidal migration, the planet actually comes close very to its host star? In some other mechanisms, it has been argued that it could be possible that the planet actually formed in situ right at the location where it is formed. So this is certainly not a solved aspect, but people are trying to look at this aspects by detecting more and more exoplanets. So we, by medium-sized telescopes, we could detect certain Jupiters which also had radius measurements from TESS mission. So TESS is a space-based transit mission, and it could list out several planetary candidates. We could do modeling by making the joint fitting of the radial velocity and the transit data all together, and by the virtue of that, we could also talk about the density of such systems. All of these systems were gaseous in nature, and when plotted around the equilibrium temperature and radius, we saw that many of the planets that we could detect by our studies had a higher temperature than the usual exoplanetary systems that were discovered previously. So as you see in this slide here, these are all the systems which were previously discovered, and the ones that are shown in red color are the ones which we conducted the studies. So planets which have a higher equilibrium temperature, they are also very interesting because they could be excellent candidates to perform atmospheric studies by ground-based studies or by space-based studies. We are already aware that missions like JWST are there in the sky, and they are trying to characterize the atmospheres of these exoplanets, which is essential to understand where the origin of these Jupiter-like planets would be present. The other bit is to identify planets if they are present around stars which are not very much old like our sun, which are not very much old like 1 giga-year-old star. So we also conducted a program where we started to look at young stars, but looking at young stars is not always rewardable. They have their own issues because they can be very active, and detecting planets around them it's not always very easy. We ended up discovering one Jupiter planet around a relatively young star. It's not the youngest star around which a planet has been discovered, but we could identify a strong lithium line in this star, and by understanding the equivalent width of line, putting it in perspective of all the previous nearby star open clusters, we could give an age limit of 27 to 625 megayears. The age error bar is pretty large, but it's definitely a star which is not 1 giga-year old. It's relatively young, and it has a exoplanet of this order of roughly 2 times that mass of Jupiter. So such studies are also very interesting to understand if Jupiters at what stage did they have, when the Jupiter formed at a stage which is very early on, then we can really say that maybe the planet was formed in situ, and it perhaps did not have enough time to really migrate from an external position to an inner position. So questions like this can be raised by performing such studies. Now, there was another interesting candidate that we discovered. So this was a planet at an orbit of 4.4 days, and it was a Jupiter-like planet that we discovered. But from test data, it had multiple sectors being observed. We started to see interesting transit time variations in the signal. So transit time variations means that normally if there is a single transiting planet, you would expect that the transit happens at the same point in time after every orbital period, but here this central transit time of the planet was also oscillating. So when we looked carefully, phase folded the test data, we found out actually there is just not the exterior planet, the Jupiter that we were talking about, but there is a hidden small Earth-like or super Earth-like planet which is present interior to this Jupiter. Now this really baffled us, because by any planetary formation models you would never expect that if you have a close-in Jupiter at 4.4 orbital day, if there could be another additional planet which is forming interior to this. So this was definitely very interesting, and a lot of efforts were gone in ground-based photometry measurements and a lot of data from medium-class telescopes, which was also complemented by precise spectrographs like HARPS in order to confirm the mass of the smallest exoplanet. So this was done in a large collaboration, and we got results and published this last year. So this was fantastic work while we were looking for a hot Jupiter, but we ended up having an additional inner planet, which was fantastic. So most of the planets that I talked about here, they have been done with modest facilities like 1 meter or 2 meter class telescopes. So this is a facility in India. So this is a place where I did my PhD from at Physical Research Laboratory. So I used the instrument Paras, and this is a place, my previous workplace in Germany, where we also have a 2 meter telescope and another Richel spectrograph placed there. So such studies can easily be done with the high resolution 60 to 70K on medium class facilities. But when we start to talk about the smallest of exoplanets, then the narrative definitely changes. Let's look at this graph where the total number of discovered planets are plotted as a function of radius. You immediately start to notice that the Jupiter-like planets, so this is in terms of Jupiter radius, they are not the bunch of planets which have highest in number. The highest number planets are actually the smallest planets. In order to confirm the smallest of exoplanets, we definitely need to do or have studies performed on these spectrographs with further refined precise radial velocity capacities. So we had a large program with HARPS and HARPS North, where we were trying to characterize the small exoplanets which had transit signatures from either K2 or TESS. So at the context of that, we discovered several of these exoplanets. So the interesting bit, so 2 of them I'm highlighting here. So while we detected the masses through the HARPS spectrograph, the radii came from the TESS. The mass and radius of one of these systems which got recently accepted, it is a very interesting, it has a Neptune-like density, almost exactly like Neptune, but it sits very close to its host star at an orbital period of roughly 7 to 8 days. Similarly, this is another system that we got out last year. This also has an orbital period of 7 to 8 days, but it has a density which is slightly higher than Neptune, and you can interestingly see what I have shown here on the, I think it's not working. Sorry about that. Okay, so on the right side of this diagram, what I am plotting is the transmission spectroscopy metric. So transmission spectroscopy metric is a metric which decides how good a particular exoplanet is if one wants to perform atmospheric characterization studies. So this is a Neptune planet, which is excellent candidate because it sits on the upper side. So this will be an excellent candidate if one wants to look at exoplanet atmospheres for a Neptune-like planet. So these are some of the studies which we could contribute from the HARPS survey that is still undergoing. So moving next, so as I mentioned previously, most of the planets that I discussed in the first half of the talk, this is concentrated around the solar-like stars. But over the last decade, the paradigm has changed. We have started to understand that not all the stars in the universe are solar-like. The most abundant stars in the galaxies are M dwarfs, and we are really avoiding a large bunch of them by not talking about them. So if you concentrate on the planet functions as a host star temperature, you start to see that most of the exoplanet detections are bundled around here. So this is the temperature of solar-like stars, and also as a virtue of log G, the surface gravity of the star, they are also very much in the solar-like star domain. So we are really not talking about systems which could be colder than sun, and also we are not talking about stars which would have a higher evolutionary status. So some of these stars are clearly not looked at with a great detail, and this is what we are trying to now compensate. So this is a plot of the spectral distribution of stars with respect to wavelength. So if you have a look at this plot, the blue part that you see is something which a HARPS-like spectrograph covers, which works in the optical regime of 380 to 680 nanometers. Now, while we were speaking about M dwarfs, the typical spectral energy distribution of a typical M dwarf, which is AD Leo here, it's shown, it mostly extends very much in the near infrared in the near infrared regime. If you're using a normal HAPS-like instrument to study stars like AD Leo, you will only be getting light in the bottom part of this curve, which means that the amount of light that you get by looking at such stars, by looking at M dwarfs with normal visible optical design spectrographs is not enough. You need a special instrument to look at this. So Caminus is one such instrument which was built with two arms, with the visible and the near infrared one, and it operates at a wavelength regime between 520 to 1710 nanometer, which is excellent to study candidates like M dwarfs. So there was a pilot program conducted at Caminus. There were guaranteed night observations. So Caminus sits on a 3.5 meter telescope in southern Spain, and it's a joint program between Germany and Spain, and it has a high resolution spectrograph which has a resolving power of 84,000, and it has two channels which work in these regimes. So the main idea of the Caminus program was to conduct a radial velocity survey of 300 M dwarfs, which means we would not know anything about these M dwarfs a priori. We do not have any information if they have planets or not, but we are going to look at and systematically search for such 300 plus M dwarfs. So this program has been running for the past 8 years and has been really successful in detection of more than 60 planets around M dwarfs. So if you happen to look at this plot where I am plotting the planet mass around the stellar mass, you see all the planets which are detected by the radial velocity method, they are plotted here. Most of the planets are concentrated in this regime which sits around one solar mass star that we also looked in the previous slide. The planets which are seen here, so as you start to see the red and blue dots which are shown here, they are the planets which are discovered by Caminus. The ones which have test follow-up, they are in blue, and the ones which came out from the blind survey, they are in red. You start to notice that a lot of this space, which was earlier not populated enough, has been able to be covered by instruments like Caminus. So definitely we need dedicated instruments in some cases to focus on particular science areas which do not have enough studies done previously, and Caminus is doing a wonderful job in that. So earlier studies, we pointed out that there are nearly more than one exoplanet or small planets, which means planets which are having a radius less than 10 Earth masses. They are present around per M dwarf for period up to 100 days. So up to 100 days, which is not very long orbit, and small planets, they are nearly more than one such planet which is present around such M dwarfs. But if you talk about giant planets which have masses greater than 100 Earth masses, they are still not a lot around M dwarfs. So the frequency of such planets is only 6%. Now if you talk about planets which exist beyond 100 days, then the frequency rises slightly. This is what we have observed with our earlier Caminus studies. The survey is still going on, so definitely planets which exist further out will be discovered later in the survey time. But based on the initial studies that we published a couple of years ago, this is something which came out of the survey. Now, what interesting things, couple of interesting discoveries that came out of the survey. So Caminus was designed to detect detect stars around the ultra cool stars, stars which will have spectral type of M6, M7, which means temperatures around or below 3000 degree Kelvin, and it was very successful in the initial operation. We could discover two planets around a star which is very, very cool, temperature less than 3000 Kelvin, Teegarden star, and it has two planets at 4.9 and 11.4 days detected early on. And this was really nice because there are only handful of systems like Proxima, Centauri system or TRAPPIST-1 system where planets have been detected at at stars which have such cool temperatures. So then there were some anomalies. We spoke about that there are not enough giant planets around M dwarfs in close orbits. That still holds true, but we could identify some giant planets in wider orbits. So there was a discovery that we did a couple of years ago around a very eccentric giant planet around an M dwarf. And interestingly, if you look at the radial velocity residuals, we see another trend which is coming up. So actually there are two planets in the system, and the second one is also currently under preparation. So there are certain interesting aspects that we also wanted to inspect So considering the distribution of small planets, they have a typical bimodal distribution. So the earlier studies with the California Kepler survey pointed out that if you start to look at small planets, which means planets which have radii between 1 to 4 Earth radii, they seem to follow a particular distribution where some of the planets, they have a peak around 1.2 Earth radius and another peak around 2.2 Earth radius. Then there is a gap of planets between these two radii. Why this exists, there are certain theories to explain this, and we wanted our way to look at such planets. So we focused on a couple of systems which would have stars having more than one planets detected from TESS mission. So here what we tried to do, we did the mass and radius diagram of such exoplanets, and we tried to place them in perspective. So what you see here is the radius which is plotted on y-axis and mass of these detected exoplanets on x-axis. The same shape planets correspond to same system, which means these two planets share the same host. The ones with the square also share the same host. What we started to notice, the planets which are present in the closer to the host star, they have rocky interiors, and the ones which are present outer to the host star, they have a relatively helium and hydrogen rich environment, so they were gaseous. So what this points out to, that maybe the radius valley exists because their photoevaporation really plays a crucial role there. By photoevaporation I mean that the host star itself is so active that a lot of stellar radiation actually blows out the atmosphere of the planet. So the planet which is located very close to its host star is just left with a naked core, whereas the planet which is at a distance away from its host star still survives with the atmosphere. So these were some of the interesting discoveries which came around. We also could identify a planet in the habitable zone of M dwarfs. So there were around half a dozen or little over half a dozen planets in the habitable zone identified with the Kamenis survey. So we could also find out, this is not with Kamenis but with HABS data, there were some interesting ultra short period planets that were coming out. So this is very interesting because this is a Mercury density, this is a planet with a density of Mercury, and this is having a very short orbital period of 7.7 hours just, and the planet mass is half that of Earth mass. So this is almost one of the smallest kind of planets that could be detected. Since this is closely located to its host star, you would expect that it holds no atmosphere, which was rightly pointed out by its high density. So such extreme detection planets are also very interesting while we make on the journey. Now, as we spoke about planets around just M dwarfs, there has also been great interest on what happens to planets around hotter type stars, which are not really formally looked by radial velocity surveys because hot stars have less atmospheric lines. In that context, we are trying to look at some stars which might be evolved, and these stars would have been predecessors, actually the hotter stars would be predecessors to these later stage evolutionary stars. So we are trying to curate a list of stars in the evolutionary stage, not really giants but subgiants. So some of this work is done by my master student R. Kaprova. He has put up a poster there, so please for details you could go and have a look. So briefly I will just mention that these are all the different kinds of planet host stars, and if you look into the planets which have been already confirmed with subgiants, the planets seem to have an orbital distribution in this way, wherein means that you would find planets which are close into the host stars but probably not so cool enough, not so close enough. So there have been, we are trying to curate a list of such stars which are subgiants, and we will be systematically conducting a radial velocity program to look at if there are planets around such stars. So this is an ongoing work. And interestingly, when we talk about detecting the smallest of exoplanets, it's not very easy because stars perform many things, and some of these things could be because of the presence of granulation or spots on their disks. And often the radial velocity variations that such processes in the star, they affect of the level of 1 meters to 10 meters per second. And in order to correct for these variations, it's very hard, and there are certain interesting bits that we try to do. We try to look at the periodograms, whatever signal that we see in the radial velocity, if it is seen in some activity sensitive lines, for example, some lines like H alpha or calcium index, if it is present in your spectrograph, we try to compare if there are such lines, and if they seem to coincide, then these lines might be produced not by the planet, but because of the activity signature. And then we also do a lot of Gaussian modeling to these lines. So if you have a planet which is detected around a star, then there's not just the signature of the planet, but the quasi-periodic variations which occur because of the presence of rotation or spots on the disk. We try to forward model it with our data and identify it. So interestingly, the period that we found out by forward modeling in the data corresponds to the same period that we found by doing long-term photometry. So there is a clear correlation that long-term photometry is giving you the rotation period of the star, which is also getting imposed on the radial velocity variations seen here. So there are a lot of things that we are doing. So in our research group, we have a visiting PhD student from Ariel University. So he is looking at solar-like stars and trying to characterize the data. So he's having some HAPS-NOT data, and he's looking at this data to correct for stellar activity variations. So there are several interesting things, and he has also put up a poster, so you could just talk to him for getting more details about it. So correcting for such variations is also very imperative. So I think I would like to summarize my talk by saying that in order to understand planetary architecture, one needs to have joint efforts while we are trying to look at massive planets by medium to on modest class facilities. It's also imperative to conduct large programs which would have a longer time baseline to characterize planets in long-term orbit, so understand the complete planetary architecture. And it's also interesting to study planets which were around stars which were not traditionally looked by, beyond solar-like stars. And definitely planets do not, detecting small planets is not very easy. Detecting them also has a lot of problems because you need to disentangle this stellar activity signal from planet signal, and that's an important research area which the current decade of astronomers are really struggling with, and a lot of algorithms are being developed for that. So with this I would like to thank and would be happy to take questions.
Thank you, Priyanka, for the nice talk. So we'll have a couple of questions. Ajit?
About light curve. So the dip in the light curve is because of the light blocked by the planet.
Yes.
Then it will also lead to microlensing, which will increase danger.
Yes.
Is that an important factor in this light curve?
So for this particular thing, we do not really bother about it, but microlensing is actually another detection method which people are looking at. So there have been some discoveries with the microlensing, but for the transit detection algorithm, we do not account for the microlensing effect. But yes, there could be reflection which is there, but it also depends upon at what levels you are able to see. So from ground-based measurements, this does not hold any case, but for space-based measurements, more accurate, these high precision photometry measurements exceed, then these corrections will be really valid.
Dip in the curve is not affected by the microlensing?
There will be an increase which does happen for microlensing events, but what I spoke about is you're only targeting when you expect the transit signature. So that's a very short amount of time that you're actually looking at. So these are two different detection techniques which are occurring hand in hand, but of course this is just to talk about transit. Yeah.
Okay, sure. Hi Priyanka, thank you for the wonderful talk. So I was very interested by the young hot Jupiter when you showed, the young hot Jupiter candidate. It's very exciting because we really don't have those. But because each error bar is quite large, I'm a bit worried about the mass measurement that you have from the RV, because for 20 million-year-old stars, we know that there's a lot of jitter in the RV.
Yes.
And we've had cases where
Yeah.
it looks like a Jupiter, we've measured the RV mass,
Yeah.
we get Jupiter mass, but when we do the atmospheric characterization in TTVs, we get a sub-Neptune-like mass. So we rule out the RVs at more than 10 sigma level of confidence. So have you tried any other follow-up methods for the system, like TTVs or something, or maybe atmospheres, to get the mass in an independent way?
So for this particular star, it at least doesn't show TTV with the data that exists with us, but definitely this is in interest to follow it up with a better instrument like HAPS. But again, getting time on HAPS is also very competitive, so we haven't really secured, but hopefully if we get in the future, then this would be something which would be really interesting to look at to reduce the error bars. You're absolutely right. Not only for the RVs, but also for the ages. This is very important.
Thank you for the nice talk. Can you please go to that slide where you had this K parameter, I think right at the beginning. There was an equation where you had this K parameter, I think. This is going to be a question from someone who's not in the field. Yes, that.
Yeah.
Yeah, exactly. So do I understand correctly that this is actually the parameter that you can measure directly?
Yes, this is a parameter that one measures. The semi-amplitude is something that you measure directly, and then you infer the mass of the planet on basis of this semi-amplitude.
So regarding that inference of the mass, if I just look at this equation, there is a degeneracy between the mass of the planet, sine iota, as well as the mass of the star.
Yes.
So how are you able to disentangle all of them and confidently say that this is a Jupiter-sized planet or a smaller one, or how do you do it?
Okay, so while making this, I did not make it quite clear. So when we are doing radial velocities, what we are getting is only the projected mass of the exoplanet. So the MP sin I things remain the same. Regarding the mass of the star, it's an excellent question because it's very important to know the mass of the star precisely in order to put a constraint on the mass of the planet. We rely on stellar evolutionary models. Given the spectral type and the other quantities that you fit, the spectral energy distribution that you have on account of the high resolution measurements that you do, you have masses of the host star coming from the stellar models. So these are only accurate up to how accurate your models are, which is something we usually planetary scientists just forget, that there's something very important. The host star might be changed, if you're really able to determine these numbers that well. So that is there. That is definitely there.
I suggest one clarification regarding the sine iota. Do you use some prior and just marginalize out or something like that? Because you don't know a priori what the inclination is.
Yeah, so we are just putting out a projected mass, and if you have transit measurements which come from anywhere, then you can take care of the inclination, because by the virtue of transit, the I can be solved. Based on the transit duration and the depth, you can solve the parameter of I. So for systems which are transiting, you would have the full mass and the radius, then only you can talk about density measurements. For the systems which have no radii estimates or no transit, you cannot definitely talk about even the inclination and nor about the radius. So most transiting systems are best when you have to talk about atmospheres or such. So yeah.
Okay, so last quick question.
Hi Priyanka, that's a nice talk. Just found a very nice question and a comment on this macrolensing. So I'm not sure the macrolensing will boost so much, because the star and the macrolensing will work depending on the distance between us and the source and the lens, and since both of them are very close to each other, how much that will affect, I'm not sure.
It does affect, because if you have so the lens is actually the star and the planet system. So if this something which is really at the background, you would have a lens. If it happens to be a single star, you would just simply see an enhancement in the light curve. But if there is a planet which is present, there will be two spikes which are present in the data. The second spike will correspond to the presence of the planet, and the distance from the main spike and the intensity of the spike will actually tell you how massive the planet is and how far it is separated from its host star. This is a standard method which is used by microlensing surveys and has led to detection of several microlensing planets. Can you please?
Can we just continue this discussion about how?
Sure.
Yeah. Because we also have a couple of announcements from the AOC. So thanks Priyanka again, and thanks all of our speakers for the sessions. And over to you for